table 7 with ticks. Let me share the table first. Okay, we have galactose, we have phospholipid, we have RNA, and we have sucrose. Now we have to tell whether these elements are present or not. Okay, what would be present in galactose? What type of molecule is galactose first of all? Is it a carbohydrate? Is it a protein or a fatty acid? It's carbohydrate. So it would have an oxygen and hydrogen, which is not given. Okay, phospholipid. What would phospholipid? Which elements are present in phospholipid? Carbon. Lipid contains carbon, hydrogen, oxygen, and if the phosphate or phospho word is used, then it means phosphorus is present. So, what is RNA? Ribonucleic acid. All. Ribose sugar and nucleic acid contain nitrogen compound, oxygen one, and phosphate linkages. Okay, sugar is again a carbon and oxygen. Okay, let's move to question number 9.2. Again, a graph is given. After Watson and Crick proposed their model of DNA, scientists investigate the possible mechanism of DNA replication. They have used so many experiments to prove it. Okay, whether the replication is a correct process or not. There was a lot of like um, fight between the, you can say, scientists on this. Two scientists grew up bacterial population, providing them with a nitrogen source containing only the heavy metal isotopes of nitrogen. As soon as the DNA is population contained N15, the scientists change the nitrogen source to one containing only the lighter isotope, that is N14. They change the nitrogen source at 0 hours. During the investigation, the scientists measure the size of population of bacterial cell. Okay, let me explain it in easy language. Now whenever we want to check if some how is some process happening, we use some isotope or radioactive isotope most of the time. Now whenever that molecule passes from a certain pathway, it emits some type of radiation which can be detected easily on an instrument. So scientists use the isotopes to check how the process is taking place. Now, what have they done? They grew a bacterial culture, and then which nitrogen source they have provided, they have given them 15, which is an heavier one, because normal is N14, nitrogen with 14 atomic number or a proton number. Now, after all the normal nitrogen is replaced with the heavier one, again they have given them the normal one. Like what would be the behavior of the normal one with this? Okay. Now, during the investigation, they recorded this reading. Okay. Sorry, can you speak louder? Okay. Then do you want me to skip this question? Because we need to. Okay, you haven't done natural logs. But you know how to use calculator. It must be given on your calculator. You can check. Okay. If you're not comfortable, we are not going doing it. Let's move to the next question then. Okay. Again, a graph is given. There's so many graphs. Okay. The question, I guess, is not right. Complete. At intervals during this investigation, the scientists remove sample of bacterial population, isolate the DNA, and measure the density of DNA. So, what do you think? Which would be heavier, the one with N15 or the one with N14? N15 one would be heavier. Okay. Now, what they have done, they have recorded this reading that generation 1 has this one, generation 2 has this density, generation 3 has this density. Okay. Now the question is, again the question is on next page. They have given us three models as well. So there is one more addition to this one. Wait, let me paste this here. Okay. So we have three possible models. Sorry. In the first model, they are saying that the in P1, let's understand the models first of all. There is a whole DNA. This would be whole replicated, which is given like this. According to 2, first of all, these strands get separated and then a new DNA is formed against them. Are you getting my point? Okay. Then in R, they are saying that only fragments, it cut into different fragments and these fragments are then copied. So these three models are given. Which of these models is separated? It's supported by the results in figure 12. What do you think so? Which model is more appropriate? Q. Because a whole new, if we look at the density of this one and this one, a whole new thing is formed. So how is this possible? Because there are two different stands which show the structure or show like shows the new model which have N15. What would be the name? It's model Q. Sorry, semi-conservative. And what would be explanation for the first unsupported model? Why do you think so that P is not the right answer? Because if we look at it, there should be two peaks, one normal and one the new one. But here we have only one. Okay, and what is the explanation for second unsupported model? Which is totally wrong. Lesser than two peaks, exactly. Because if we look at these results, these are totally like each one of the offspring or the third generation has some part of the parent one from generation 0. Okay. Describe the structure of DNA would be quite easy for you. Okay, what are the basic units of the DNA? Okay, okay, okay. So what is the basic unit? What is the basic unit? What basic unit of DNA? What do we call the basic unit for DNA? For example, let me give you an example. Like the basic unit of protein is amino acid. The basic unit of fats is lipids are fatty acids and glycerol. So what is the basic unit of DNA? Nucleotide. So very first point should be this. Then we have to mention that there is a phosphate backbone. But how is this formed? Phosphodiester bond. This you have missed. Phosphodiester bond, and it is formed between nucleotides. Okay. Then there are different type of nucleotides. Means the adenine, the thymine, guanine, and cytosine. Okay. Then we have told the base pairing as well, that which bonds are formed. Can you tell me which bonds are formed between A and T and which are formed between G and C? Hydrogen bond. So we have a double hydrogen bond with AT, and between GC we have a triple one. Okay. So a double helical structure is formed by which type of bond? What causes it to turn to form a helical structure? Hydrogen bond. So these points needed to be written because these are very very important points. Okay, next question. Name and describe five ways substances can move across the cell surface membrane into the cell. Okay, so the very first that you have told is diffusion. That is a very basic movement by which the molecules move from high concentration to low concentration or down the concentration gradient. Second, you have mentioned the osmosis, the movement of water molecule down the concentration gradient, water potential. Number three, you have mentioned the facilitated diffusion, I guess, that that is actually carried out by the carrier proteins. Okay, fourth one you have mentioned the active transport that is against the concentration gradient with the use of energy from ATP. And the last one, what did you mention? Sorry, I didn't get it. Your voice is so low. I'm not able to hear it. Co-transport. Okay. The co-transport, for example, if one molecule is moving or passing through the carrier protein, the other molecule can also bind to that protein and move inside. Okay. Let's move to the last part, I hope. So we have two cells, cell A and cell B, and their magnification is given. And one is animal cell and one is prokaryotic cell. Okay, what do you think is the animal cell and what do you think is the prokaryotic cell? Here is a diagram. So this must be animal cell and B is prokaryotic. Okay. Okay. Linear DNA. Okay, so the very first difference is about the magnification. The magnification of cell B is more as compared to the third one. So it means that the cell A is bigger as compared to cell B. Then cell A has a nucleus while B has free or a floating DNA. Then A has mitochondria while it's not observed in B. Golgi or the apparatus is observed in A when not in B. A capsule is not there in A but in B. Uh, for a complete round, would you have said that circular DNA? You can write that, but I couldn't see that one in this diagram. So it's better not to mention that if you cannot, like, it's not that much visible. You can skip that. What else can be? One thing you have also told, it's totally skipped my mind. Is it about the like histone protein or something? Have you told something about that? Okay. So the A has the DNA bound to the histone protein while B has no such thing. Okay. So this is all for this paper.